Okay, hi class. Welcome to our last Zoom review. We're going to go over Chapter 8 and the final. This was supposed to be a live session, but something came up and I had to pre-record it. But we're going to basically do the same thing. So I'm going to pull up the final exam review. We're going to go over the main points from each chapter, and we'll just kind of break down everything. But first, let's talk about your final exam. Your final exam is on Saturday, December 5th, and it's open for 24 hours. So it's open all day. So literally midnight on Friday, you will be able to start the final exam because technically that is Saturday. So Saturday, that's kind of when it starts, and then you have the entire day Saturday, and it closes at 11:59 p.m. on Saturday night. So yeah, there's your 24 hours. Please find a time in that time frame to take the exam. If you have any issues while taking your final exam, please let me know, like if the exam shuts you out or you lose Wi-Fi connection, anything like that, I will be on call to open or reopen that exam for you. So yeah, let's get started with going over our exam review, which has been posted on Blackboard for the last week or two. So yeah, let me just kind of share my screen and get started with all of that. Okay. All right, so as you can see, this is a comprehensive final. So it is going to be over everything, not everything, but all the chapters that we have covered. So first, I kind of did a breakdown of what your questions are comprised of. So there are no fill in the blank questions. Y'all are probably so happy for that I don't have to go back in and recorrect those answers that were already correct. But yeah, so 37 multiple choice and 13 true false. So chapter 2 through 3, let's just kind of talk. I'm not going to pull up the PowerPoints for every single chapter, but I will pull it up for chapter 8. But let's look at chapter 2 through 3. What are those main questions? So there's roughly going to be, there's 50 questions total, roughly 5 or so questions from each chapters 2 through 7. But yeah, let's just kind of start. So what do you have to recall from chapter 2 and chapter 3? So first, it's going to be the energy systems. That was the main bulk of those chapters. So understanding the ATP PC system, just knowing how, when is the system active, what kind of exercise is going to stimulate this system, what enzyme is used in the ATP PC system, just kind of information, general information about that energy system. Glycolysis, we went a lot more in depth with that. So you have to know the main enzymes that we talked about in glycolysis. You have to know how much ATP is produced from one molecule of glucose in glycolysis, how many NADHs are produced there, what is the end result of glycolysis. So those are just kind of the main points there. Oxidative metabolism, you have to know that is comprised of the electron transport chain and the Krebs cycle. Knowing how much ATP is produced in the Krebs cycle, knowing what are the other byproducts produced in that cycle, also knowing the electron transport chain, how does that system work, just kind of the basics of that system, and then by the end of the electron transport chain, knowing how many ATP are produced from one molecule of glucose or from one molecule of glycogen. Also knowing what is the final electrons acceptor at the very end of the ETC. So yeah, just kind of knowing basics of the 3 energy systems. When are they used? What's their primary function? And what are the byproducts that are produced? Next here is looking at those enzymes that I mentioned kind of earlier. So phosphofructokinase, knowing that that's the rate limiting enzyme in glycolysis, knowing that creatine kinase is in the phosphagen or the ATP-PC system, knowing what is the primary role of creatine kinase is going to be important. Lactate dehydrogenase, knowing that that's the enzyme that takes lactate, converts it to pyruvate, or pyruvate to lactate, back and forth there. And I kind of already mentioned ATP production from each energy system. So yeah, just kind of basics of that. One thing I'll mention as I'm going through this is you will see questions that you had on exam 1 and exam 2 and exam 3. So my questions, I reuse those for the final. There are a few new ones added in there, but a lot of them you've already seen before. Okay, so chapter 4 and chapter 5. So what did we primarily learn in chapter 4? We learned a lot about the integrity of skeletal muscle. So knowing what makes up a sarcomere, so kind of knowing about that sarcomere, knowing what non-contractile proteins are, knowing what contractile proteins are, structural proteins, which we know actin, myosin, titin, troponin, tropomyosin. So knowing all those structures in the sarcomere. And then that gets us to the sliding filament theory. So knowing those steps in the sliding filament theory, what exactly is happening, how does the muscle produce force? Next, we talked a lot about type 1 versus type 2 fibers. So knowing basic characteristics of a type 1 fiber versus a type 2 fiber. There, you also had a little bit of information about different muscle actions. So knowing about concentric, eccentric, isometric, that's also going to, or you should know that. Next, moving on to chapter 5, we talked about the action potential a lot. We talked about depolarization and repolarization. So knowing basically the steps from resting membrane potential. So knowing the millivolts of resting membrane potential, knowing the threshold level of activation, that negative 55 millivolts. So negative 70, negative 55, and then knowing depolarization, the millivolt changes by 100 to positive 30 millivolts during depolarization. And then we have repolarization. So just kind of knowing what happens to sodium and potassium during repolarization, depolarization, and yeah, so just kind of knowing basics of what happens there. Sensory versus motor neurons. So how do you need to know this? First, you do need to know a little bit about motor units, knowing that that is an alpha motor neuron, and then knowing how that motor neuron controls the motor unit, which comprises of all those muscle fibers, but then also those sensory neurons as well. So how the sensory neurons sends that information to the spinal cord, where we process it sometimes with an interneuron, and then send it back with a motor neuron, and that causes the action. And the next is sympathetic versus parasympathetic system, knowing what chemicals are involved. So what chemicals are secreted when the sympathetic system is activated versus parasympathetic. Know what are the primary roles, or what happens with sympathetic. So what's going to happen to blood flow? What happens to glucose and glycogen? What's going to happen to heart rate and blood pressure versus parasympathetic? So just kind of knowing the difference between those systems there. Chapter 6, main things we talked about. Number 1, blood flow through the heart. So knowing that pathway. So if I say that blood is entering into the right atrium, you have to know, where did that blood just come from? And then where is that blood going from there? Also, you have to know, is that blood oxygenated or deoxygenated? So yeah, just kind of knowing, if I give you a place in the heart, you have to know, like I said, where did it come from? Where is it going? Oxygenated, deoxygenated. So knowing peripheral versus venous blood, knowing that as well. Arterial versus venous blood, peripheral versus pulmonary blood. So just kind of knowing everything there is about blood flow through the heart and through the body. Another big factor we talked about was stroke volume and what factors increase stroke volume. You're going to have a handful of questions about this as well. So knowing about PV, that's plasma volume. So knowing if we have an increase in plasma volume, how does that affect stroke volume? Well, it's going to increase it. But plasma volume is going to increase BV, that's blood volume. So now we have larger blood volume. That's going to return back to the heart, and FSM is the Frank-Starling mechanism, having to know that with increased muscular contractions, we're going to have increased blood flow back to the heart. That's increase in venous return. So with an increase in venous return, it's going to stretch those ventricular fibers. So when those fibers stretch, that's going to cause an increase in contractile force. So first, with the stretch, that's in diastolic volume, EDV increases due to the increase in Frank-Starling mechanism. And then as those ventricular fibers stretch, they're going to contract. And that is going to increase ESV in systolic volume, which is the amount of blood left over. Sorry, it's going to decrease ESV, which is the amount of blood left over. And then stroke volume is going to increase in there too. This may be one of the trickiest concepts, is understanding the factors, those cardiac, what am I trying to say, terms. So just knowing the definition of those first, and then knowing the relationship between all of those. So go back and watch, re-listen to the chapter 6 lecture. I find that was my favorite lecture. But yeah, if you have a question on that stuff, always reach out to me as well. Okay, but another main topic was intrinsic versus extrinsic control of the heart. We know intrinsic control is autorhythmicity. It's the ability of the heart to control its own impulses, and that is regulated by the SA node and the AV node and the Purkinje fibers. So just kind of knowing intrinsic control from within the heart, that's where we see those impulses. And then extrinsic control is from outside of the heart. So the medulla oblongata also has extrinsic control over heart rate, blood pressure, kind of all of that. So going back and looking at what chemicals are released, and this is where the sympathetic parasympathetic comes in, where it has control on the heart. So just knowing the difference between those two factors. Blood pressure throughout the body, knowing where it's highest in the body, knowing where it's lowest in the body, knowing what factors are going to increase blood pressure or decrease blood pressure. Know that there's a relationship between pressure and resistance. If there's an increase in resistance, there's going to be an increase in pressure. Also know that the increase in a pressure difference is going to allow for an increase in flow. So if we have a greater area or difference in pressure between one area to another, that's going to increase blood flow. So yeah, just kind of knowing that factor, kind of those factors there that have to go with blood pressure and blood flow. So next right here we have terminology. Autoregulation is the regulation of blood flow. So our blood vessels have the ability to constrict and dilate, and that's due to hormones, but it also has to do with the vessels themselves. So they can control themselves. That's autoregulation. Autorhythmicity, I just went over with intrinsic control. Ejection fraction, so going back and looking at the definition for ejection fraction and what is considered a normal ejection fraction value. And then AVO2 difference, so arterial venous difference or oxygen difference. I spend a little bit of time going over that in the lecture. So knowing basically the main definition of it, and then how does it change with exercise. So the difference in oxygenated blood or oxygen in the blood, how does that change from rest to exercise? And then last for chapter 6 is training adaptations. So if I am considered a trained athlete, probably aerobically trained, what changes have I seen to my cardiovascular system? What changes have taken place to make me a better aerobic athlete? So that's towards the end of the lecture, specifically this table right here. Okay, moving on to chapter 7. So chapter 7, one of the main things we want to focus on here is inspiration versus expiration. So knowing the muscles involved in inspiration and expiration, and then also knowing the difference between pressure and volume, so those inverse relationship. So as the volume of the respiratory cavity increases, the pressure in the respiratory cavity decreases, and vice versa. That's Boyle's law. So knowing those laws is really important. So Henry, Fick, Boyle, and Dalton's law. So knowing all those laws, that's another big one too. Factors that influence gas exchange or gas diffusion. So this is how easily is oxygen and CO2 able to cross a membrane. So what factors influence that? So we know that when we're talking about in the lungs, we know that the surface area of the alveoli. So knowing that we have a tremendous surface area and all those alveoli are wrapped in capillaries. Also knowing the thinness of that respiratory membrane, it's really small and it's permeable, so those gases can easily diffuse across. And then lastly is the driving pressure, the driving force. That part's also a little tricky to understand, is knowing how many meters of mercury or millimeters of mercury is needed to push oxygen across the capillary membrane. I think it was 65 millimeters of mercury is needed for oxygen and only 6 needed for carbon dioxide. But yeah, that's just the pressure. It's just like the driving force that pushes it across that membrane into that certain area. Yeah, so knowing about those factors. Partial pressure of oxygen. Oh, I just went over it. The driving force for each one. So you do have to know that. Like I said, that's a tricky part. If you need help relearning that, just reach out to me. Partial pressure of oxygen and CO2, alveoli tissues, that driving force. Go back and listen to that lecture. I think I explained it pretty well in the actual chapter 7 lecture. So re-listen to that or reach out if you want more information, but that will be on there. Next, the last big thing we talked about was the oxyhemoglobin dissociation curve. So we know that red blood cells carry hemoglobin, or they have hemoglobin in them, and then the hemoglobin binds the oxygen. We know that hemoglobin can bind 4 oxygens, and we know that when hemoglobin lets go of the oxygen, it means it's delivering it to the tissues, which is a good thing, especially during exercise. You need to know what factors are going to shift that curve to where hemoglobin lets go of the oxygen and it's delivered to our working muscle. So what were those factors? Temperature. So as the temperature of the blood increases, or just our entire body temperature increases, that's going to shift the curve to the right. When it shifts to the right, that means that hemoglobin is less saturated with oxygen, which is good because it lets go of it. So yeah, shifts over and loses its oxygen, which gives it to our working muscle. So temperature. Acidity is the other one. Especially with anaerobic exercise, acidity is going to increase. And then the Bohr effect occurs where the hydrogen ion then binds onto the red blood cell, preventing oxygen from binding. And so that's how oxygen lets go, and it lets go at our muscles. And then last was the byproduct of anaerobic metabolism, which is 2,3-DPG. And that also will shift the curve. So yeah, knowing what those 3 factors are, and then knowing just what does it mean when that curve shifts, and I just explained it. So terms, just basic terminology, just like you had on the chapter 7 quiz, you had a lot of fill in the blanks on those. Now you'll just have multiple choice on those highlighted terms right there. All right, so now let's get to chapter 8. So I'm actually going to pull up the PowerPoint slides and we'll kind of just go over this together. But yeah, so I'm going to stop sharing this one and I'll move and I'll switch over to the PowerPoint. Let's share chapter 8. Okay. And I just took a picture of the review for my phone so I can just keep it on the screen for me and make it easy. If you need to follow along on your review, please do so. Okay, so the first thing is really going to be the structure of hormones, kind of what are they made of? And you had quiz questions on your quiz about this as well. So knowing that peptides are made of amino acids, steroids are made from cholesterol, and then modified amino acids are also contained of amino acids, but specifically tyrosine and tryptophan. So yeah, just knowing that, which you already had a question over it, so should be good. And then you also had questions over this, but you'll see it again. Hormone release. So there are 3 different ways that hormones are released into our body. And so endocrine, we know, secreted into the bloodstream. Paracrine, released and just kind of travels to the cell that's right next to it. And then autocrine acts on itself, or it's released and then it reactivates wherever it came from. So yeah, you already had a question over that, but be familiar with it because you'll see those 3 terms again. Okay, next is well, let's just kind of look at this and see. These were just examples of autocrine, paracrine signaling. You may have a question about which of the following uses an autocrine signaling pathway, or which of the following uses a paracrine signaling. So yeah, you can go and look at that. Downregulation, upregulation. You may have a question. You had a question about downregulation on your quiz, so you may have a question about upregulation. And then knowing about those peptide receptors. So, and again, you already had a question. I keep saying that. I feel like I'm saying that over and over again. But just be familiar with how those receptors work on the DNA, which are kind of listed on these 2 slides here. I know for a fact that you do not have a question over this slide here or this slide here, just on those previous ones. What you will have a question on is feedback loops. So positive versus negative feedback loops. We talked about this in chapter 5, and the body kind of relies on these feedback loops to return us to homeostasis or equilibrium. So just kind of knowing about negative versus positive, first the definition, and then maybe an example. And I give those examples right there. All right, so on to part 2. Let's see. Yes, you will have questions about half-life and pulsatility. You did have a question on your last quiz, or chapter 8 quiz, about pulsatility. So you'll probably have a question on your final exam about half-life. So first, what's the definition of it? And then how do we create a longer half-life? You can see here at the bottom in my notes, hormones bind on to another protein or a molecule, and that's how they extend their half-life. And then how does a hormone degrade or break down? And you can see also in my notes, with time or receptor interaction, that's how the hormone will break down and degrade. So you may have to know those details. Yeah, so you'll have a question over those. And then you wrote about circadian rhythm on your assignment, and you did have a question about the pineal gland and melatonin on your quiz, but you may see it again. Also knowing these 3 factors, what factors influence circadian rhythm. So yeah, just stuff like that. All right, so next we want to talk about the hypothalamus and pituitary axis. You'll have a question about this on your exam, just knowing that these 2 structures in the brain work together to influence hormonal release. They influence when those endocrine hormones are, or those endocrine glands are activated, and when those hormones are released. So just knowing that these 2 structures work together. You may also have a question about the anterior pituitary versus the posterior pituitary. And I think you may have had one on your quiz as well. Knowing that the pituitary only releases 2 hormones, where anterior releases about 7 or so. Next is thyroid releasing hormone. You wrote about this on your assignment. You wrote about how the hypothalamus kind of influences the release of these thyroid hormones. So just kind of being familiar with that. You already wrote about it, so should be well versed. And then knowing about T3 and T4, that those are the actual thyroid hormones. And then we also have calcitonin, and what the purpose is of this. You you had a question about calcitonin on your chapter 8 quiz. So you probably won't see another one about calcitonin, but you may see one about parathyroid hormone. And a few of you did get confused with this one, and you put PTH instead of calcitonin. They are very similar because they both work with calcium, but calcitonin blocks the release of calcium from bone and other structures, where parathyroid hormone stimulates the release of calcium from the certain structures, and it increases the plasma concentration of calcium, which means it's in the blood. And this picture here kind of shows that. So you may have a question about PTH on your final. Looking at my review. So yeah, we already went over the hypothalamus pituitary axis, parathyroid hormone, calcitonin, knowing about the thyroid hormones. And that gets us to growth hormone. So growth hormone, it's just kind of knowing statements about growth hormone, knowing just kind of like what are the main roles. Growth hormone stimulates protein synthesis. It permits the accumulation of lipids. It prefers to use fat for fuel when it's released. So yeah, just kind of knowing a little bit about that. And then also knowing circadian rhythm of growth hormone, knowing that it is highest at night, not specifically at night, but when we sleep. So I guess if we slept during the day, like this study shows, or that graph shows at the bottom, it's going to be highest then. So it's mainly highest at sleep. Most of us do sleep at night, so it's probably going to be highest at night. And also it has a kind of repairing function where it helps to repair all of our tissues. That's why when we sleep, we're resting. Growth hormone works to repair our muscle tissue or damaged tissue, help us recover. Okay, next is IGF, so insulin-like growth factor. So just kind of knowing some basic facts about this one, knowing that it actually does have all three, or it can use all three release patterns. So endocrine, paracrine, autocrine, it kind of functions and it uses all of those. It also has 6 binding proteins, which this picture shows here. Purpose is to transport IGF to the target cell. So we want to extend its half-life, which allows it to be more potent. So it's more potent in the blood, and it can live for a longer period of time before it degrades. This goes into more about IGF. It's helpful for muscle building, cell rejuvenation. It also works with muscle or bone growth as well. Next is testosterone. So just kind of know the basics of it. So what are the primary purposes of testosterone? What does it do? And then what happens to testosterone with exercise? I went over this in the lecture. So we know it's increased acutely with exercise, but what about chronically? So what about chronic resistance athletes or chronic endurance athletes? How does testosterone, what is going to be the difference between those two types of training? So go back and listen to the lecture if you don't remember that. And then also about the diurnal rhythm, so circadian rhythm of testosterone, it's going to be highest in the morning. Next is POMC peptides, which is just a class of peptide hormones. The one you had on your quiz was beta endorphin. So you'll probably be asked about these other two ones, so ACTH and MSH. I go over those two here. So this slide will be important. Just knowing what is MSH, what's the main role of it, which you see those roles there. And then knowing that adrenocorticotropin, ACTH, works with cortisol. So it stimulates cortisol production. So yeah, just knowing the basics of those hormones. You were already asked about beta endorphin, so you probably won't be asked about it again. Next, adrenal gland. You had to know on your last quiz that the adrenal gland was the fight or flight gland. Now you have to know a little bit more about the hormones that are released from this structure. So I do know that there is a question on the exam about these right here. So knowing that aldosterone is a mineral corticoid, and then knowing that cortisol is a glucocorticoid, and then knowing that androgen and estrogen are sex steroids. So you do have to know that. That's big. So just knowing what class, basically the classes of these adrenal hormones, you have to be familiar with. And then epinephrine and norepinephrine, you have to know that those are catecholamines. They are catecholamines. That's their class. And we know that catecholamines are organic nitrogen components. They're amino acids. So yeah, you have to know the classes of hormones. And then the role of epinephrine and norepinephrine, which is talked about right here. What's the role? What happens when they're released? So we have 2 slides that just kind of go over those main roles. You already had your chapter 8 quiz, so you don't need that anymore. But continuing, now let's talk about aldosterone. So knowing aldosterone mainly works with water balance. It helps to regulate blood pressure and influences the acid-base balance. It stimulates ADH, which is arginine vasopressin. Again, just know the main roles of it. You may have a question about antidiuretic hormone, which is ADH, which is the same thing. You do have to know this. It's the same thing. Arginine vasopressin, ADH is antidiuretic hormone. So this right here is the same thing as what's on this next slide here. This is that hormone, arginine vasopressin. Know that it's very similar to aldosterone. They work with water loss, plasma volume. So these 2 hormones are going to be very important with aerobic athletes or athletes who just sweat a lot and lose water. You don't have to know about this slide here, but you do have to know about cortisol. You had a question on your your quiz about primary catabolic hormone in the body. The answer was cortisol. Now you'll probably have more questions about cortisol, such as, what are the main roles here? So stimulating free fatty acid mobilization from tissues, also amino acids, blocks entry of glucose in the cells. I go over a lot or more detail than I am now on the chapter 8 actual lecture. So if you need to understand cortisol more and understand those 3 bullet points, go back and listen to that. Also, the circadian rhythm, cortisol is highest in the morning. It doesn't influence our sleep and wake cycles, but like melatonin does. But it does help us to become more alert. So cortisol works in the morning, and it also responds to low glucose levels. That's also when it gets high. So yeah, you may have a question about that. Next is looking at adipocyte hormones. So adipocytes are fat cells. So fat cells, we have leptin and adiponectin. So we can see here leptin versus adiponectin. Again, I'm going to say it over and over again, is just know the basics that from each hormone. So know the role of leptin and know the role of adiponectin. And then what happens with obesity? So what happens with obesity to the leptin levels and the adiponectin levels? So it kind of says right here the basics of all of this. I think in the lecture I go over into why is leptin higher in obese people, or why is adiponectin lower. So I spend time in the lecture going over all of those details. All right, getting towards the end here. Last is pancreatic hormones. So knowing the difference between beta cells, alpha cells, delta cells, and PP cells. Again, just knowing the definition, but you may have some practical questions here. So like if we know the role of beta cells, they secrete insulin to lower blood glucose when it's high. So I may ask you a question like, okay, this person just ate a very large meal, mainly of carbohydrates, and now they're just sitting on the couch and they're not exercising. Well, blood glucose is going to be very high. And if we're not exercising, that's not going to decrease it. So the body will secrete beta cells. So you will have some practical questions like that, kind of from the whole lecture. Like if this is going on, what hormone is most likely to be released? So yeah, just kind of be familiar with definitions. And then if you know the definition, you're more likely to be able to answer a practical question, an applied question. So be prepared for applied questions that challenge your knowledge. And then lastly, with pancreatic hormones, here's kind of another example of when we would see an applied question. So with exercise, blood insulin concentrations will decrease. That's going to stimulate the liver to release what? So I answer that question in the lecture. But yeah, so just kind of knowing, like, here's another one. If I ingest carbs or protein, work out, what happens to insulin? We see right here, insulin's going to increase. It's going to increase to get those fuel sources into the cells so we can use them for exercise or recovery. So yeah, last here is your last quiz. I know people aren't very happy about this last quiz, which is on Wednesday. What's Wednesday? December 2nd. And it is open until midnight. So you have the entire day on Wednesday to take this quiz. Here's your review. This quiz prepares you for the final. Since the final is comprehensive, so is this quiz. All right, so looking at formation or breakdown of glucose or glycogen. So formation versus breakdown. Formation, look for the root word genesis, means formation or creation. And then breakdown, look for lysis. Lysis is breakdown. So like gluconeogenesis, that would be formation of a glucose source or glucose or carb from a non-carb source. Or glycogenolysis is breakdown. So again, just look for those root words, genesis versus lysis. We already talked about this, the products and end results. So what is produced in each of those energy systems and what's the final end result of those energy systems? Here, when I'm exercising versus at rest, what % of my energy is coming from fat or coming from carbs or protein? So maximal exercise versus rest, what is the percentage of the fuel sources that are contributing to my ATP production? Structures in the sarcomere, so we already know actin, myosin, all those other contractile or non-contractile proteins. Muscle protein interaction and force production, so this is referring to the sliding filament theory. Resting membrane potential, threshold level of stimulus, knowing the negative 70 millivolts versus negative 55 millivolts. So knowing about the action potential. Here, this is chapter 6. It looks like I'm going to give you, blood is coming from here and about to enter into here. Is it venous, arterial, pulmonary, peripheral, oxygenated, or deoxygenated? Factors that increase stroke volume, already went over all of those. There's a lot of them. Oxyhemoglobin dissociation curve, what factors shift that curve and what does that shift mean? Gas diffusion at the lungs, what are the 3 factors that increase or influence rate of diffusion of oxygen? And then brain structures involved in the hormone release. So that's going to be the pituitary gland and hypothalamus. So that's your chapter 2 through 8 quiz, and that's the review. Like I said, that quiz is on Wednesday, December 2nd until midnight. So what else was I going to talk about here? Give me just a second to kind of look and see what else I have. Oh, I know what I was going to talk about. Oops, not that. Okay, so the course instructor evaluation. So that's what I kind of wanted to mention is if you haven't done it already, it's really helpful for me to know what you liked and what you didn't like about this class. I am teaching this class in the spring semester. I'm teaching 5 sections of it. They are smaller classes. But yeah, I love your feedback. Just know this class is not meant to be taught online. I am teaching it in person. So hopefully this is the last time XPhys is taught online. I hope it was an easy, smooth class for you. I know online settings are sometimes difficult, but I really hope you took something from this class and carried it with you because you're about to take applied XPhys. So that's your next, and you have to maintain or you have to retain this information that you got here. And so I hope I was a good enough teacher, and I hope this class worked for you. But yeah, so I wish I got to meet you in person, but I enjoyed being your online instructor. So if you have any questions, please reach out to me before December 5th. That will be your final exam. And like I said in the previous email, that I will have final grades submitted by Monday, December 7th. So that means at midnight, once it turns into Tuesday, those grades are finalized. So, I mean, yes, I can ask for a grade change, but I rarely like to do that. So all of the questions about grades, I will not answer after Monday. So Monday is the last time for you to reach out to me and talk to me about your grades. So please do that before then. And the extra credit, that's the last thing I wanted to say. The extra credit is very important for you. It doesn't matter how many questions you answer on there, as long as you submit it to me with something done, and I can see that you gave a little bit of effort there, you will get bonus points. And that's what a lot of you need, is those bonus points. So please just try to answer those questions, and it will help you prepare and study for the final. That's also due on Wednesday. So you have 2 more things to do before the end of this semester, and then take your final. So that's all I have for you. If you have questions, please reach out to me. I love hearing from you. But yeah, I hope you'll have a great winter holiday and stay COVID safe. See you.